Myself Dr. Sunita Patra, Assistant Professor, Department of Botany from Government Nagarjun Postgraduate College of Science, Raipur, and now I am going to tell about tertiary structure of proteins. So, again there is a disclaimer that I duly acknowledge all the known and unknown sources from where I have taken the information and slide, and I have taken and compiled this information for the study purpose. So, I duly acknowledge all the known and unknown sources. Now, coming to the tertiary structure of protein. Tertiary structure refers to the complete three-dimensional structure of polypeptides, unit of a given protein. So, polypeptide, when it is arranged in a three-dimensional or 3D pattern, it is known as tertiary structure of protein. Now, this you can see the tertiary structure of protein defines specific all over 3D shape of the protein, 3D shape or 3D structure. Tertiary structure is the base of various type of interactions between the domains, the side chains, the polypeptide chain, and there are so many several forces which are involved in the formation of this 3D structure of protein. So, as tertiary structure stabilization in globular protein, tertiary structure frequently stabilized by hydrophobic amino acid residues in the protein and also in the secreted. That means, the tertiary structure, the stabilization is different in globular protein and in secreted proteins. The tertiary structure are frequently stabilized in globular protein. It is stabilized by hydrophobic amino acid in the protein core, and in secretory proteins disulfide bonds between the cysteine residues. It helps to maintain the protein's tertiary structure in the secreted proteins. Now, this is the structure. There are too many shapes, sizes, motifs and domains which we will see. Interactions. What are the forces, driving forces which actually stabilizes the tertiary or which gives the 3D structure of the protein? So, there are these bonds, they are all the weaker bonds except the disulfide linkages. So, interaction stabilizing tertiary structure is disulfide bonds, hydrophobic interactions, hydrogen bonds, ionic interactions and van der Waals forces. So, we will see one by one how these forces and how these bonds they are formed together to give the protein a three-dimensional structure. Now, coming to the disulfide linkages. So, cysteine amino acid, it has a sulfur molecules. So, when two cysteine molecules they come together, they form a linkage. So, this linkage, it is known as this bond formation, it is known as a disulfide bond, a covalent bond between sulfur atoms of two cysteine amino acids. If there yes, we can see there is a formation of a loop, and this loop is formed due to the two cysteine molecules when they come together, and there is formation of a disulfide linkage between these two cysteine amino acids. And this again we can see the disulfide bond formation. There are two cysteine residues, amino acids, and then formation, oxidation and reduction of disulfide linkages. You can see there the formation, the bond formation between two sulfide atom of two cysteine molecules. They form a bond which is known as a disulfide bond. Now, coming to hydrogen bonds. The hydrogen bonds, they are the weaker bonds. The bonds are weak, and we can easily see the formation. The red color in the bond is the structure which is formed. It is the hydrogen bond between which gives the protein a three-dimensional structure. It also contributes the formation of three-dimensional structure in bond. These bonds are very weak, which allow to be broken easily. Now, hydrogen bond occurs not only between the polypeptide chain, but with the surrounding aqueous atom. That means, if the protein it is forming a three-dimensional structure, so hydrogen bonds can be formed within the polypeptide itself and also between the surrounding aqueous medium. Now, what are the forces controlling? So, one of the is hydrogen bonds which we are discussing. And now, how these hydrogen bonds they are formed? Polypeptides contains numerous proton donors and acceptors, both in their backbone and in the R group of the amino acids, where we have seen in the basic structure of the amino acid that amino acid it has a amino group, a carboxyl group, hydrogen and R group. So, polypeptide had a numerous number of proton donors and acceptors in the backbone and in the R group of the amino acid, which contribute to the formation of hydrogen bonds. And the environment in which the proteins are formed, they also contain the hydrogen bond donors and the acceptors of the water molecules. So, all these molecules, they actually contribute the formation of hydrogen bonds. So, you can see here the formation of hydrogen bonds, how they are formed between the serine residue, here it is, and so there should be a donor and then there should be acceptor and formation of a very weak bond, hydrogen bonds. Now, the third force which is which actually enforce this three dimensional structure of protein is hydrophobic interaction. Proteins are composed of amino acids and that contain their hydrophilic or hydrophobic R group. That means, we know that proteins are composed of different amino acids and different amino acids had hydrophilic nature or hydrophobic, and this hydrophilic or the hydrophobic nature is due to the different type of R groups which are present in different amino acids. Each amino acid has different R group. So, the proteins composing of different type of hydrophilic, either hydrophilic or hydrophobic R group, this actually contribute towards the hydrophobic interaction. It is the nature of the interaction of different R group with the aqueous environment that plays the major role in shaping protein structure. So, once we have seen the what are the different type of forces. So, one was disulfide linkages, then second was the hydrogen bonds which has been formed, and again it is the hydrophobic interaction which plays a major role in shaping protein structure. Now, the spontaneous folded state of globular protein is a balance between the opposing energetics of hydrogen bond between hydrophilic R group and the aqueous environment and the repulsion from the aqueous environment by the hydrophobic R group. So, there are two type of groups, hydrophilic R groups and hydrophobic R groups, and there is a balance between the opposing energetics of hydrogen bonds between hydrophilic R group and the aqueous environment, and also repulsion if the group R group is hydrophobic in nature. So, repulsion from the aqueous environment by the hydrophobic R group. So, this is the balance between the hydrophobic and the hydrophilic R group found in different type of proteins. The hydrophobicity of certain amino acids R group will drive them away from the exterior group into the interior if the characteristic is of hydrophobic nature. This driving force restricts the available conformation into which a protein may fold. So, this is actually the nature and the driving force which restricts the available conformation into which a protein may fold. So, hydrophobic nature is due to the R group, different R group present in different amino acid, and then hydrophobicity and the hydrophilic and the hydrophobic nature of that particular amino acid. Now, electrostatic forces, or what we can say, electrostatic forces are mainly of three types. Electroforces process are charge to charge, charge to dipole and dipole to dipole. Typical charge to charge interactions that favor protein foldings are those between oppositely charged R groups such as K or R or D or E. That means, these are the different nomenclature given to the different type of amino acid. It is a universally accepted nomenclature with alphabets which denotes a particular type of amino acid. So, electrostatic force typically charge to charge interaction that favors protein folding, charge to charge are those between opposite charge groups. Suppose if the both of the groups they are oppositely charged, they will favor the interaction and also favor the protein folding, such as the opposite groups such as K or R or D or E amino acid, they are oppositely charged. So, they will favor the interaction and the folding, and which will lead to three dimensional structure of the protein. Now, charge dipole interaction. Charge dipole interaction, it actually refers to the interaction of ionized R groups of amino acids with the dipole of the water molecule. Actually, we are also seeing that the bond formation is also influenced by between the amino acids and also with the environment, aqueous environment surrounding the protein. So, the charge dipole interaction, it is the interaction between the R group of amino acid with the dipole of the water molecule in the surroundings. So, here the charge dipole interaction comes, it influences formation of 3-dimensional structure of protein. Now, here we can see the ionic bond formation. So, it is the residue from aspartic acid, and the bond formation here is from lysine and the aspartic. So, residue from lysine and the residue for aspartic acid, they come closer and move towards the formation of ionic bond. So, here we can see the ion from R group, and this R group, they are different for different amino acids, and they form the salt bridges through the ionic bond. Now, the figure shows the formation of salt bridges. The amino and the carbonyl areas of the protein attract and form ionic bonds. So, there are disulfide linkages, there are hydrogen bonds, there are hydrophobic linkages, electrostatic bonds and salt bridges or the ionic bonds which are formed between NH and COO area of the protein. They attract each other and they form ionic bonds. Now, hydrophobic forces. Now, there are another forces which drive the protein and it gives the final 3-dimensional structure of the protein. So, you can see the hydrophobic forces which are involved, they are close attractive, they are all these bonds, they are very weak, but they altogether they form and give a protein a 3-dimensional structure. So, hydrophobic forces, they are also a very weak bond, weak, but collectively collective interaction gives a large area a very stabilized structure. All these weak bonds, they are weak bonds, but when they are collectively impressed, they are forced the interaction of different bonds, they give a very stabilized structure. Van der Waals forces. Van der Waals forces are both attractive and repulsive van der Waals forces that control protein foldings. That means, there are two forces, attractive forces and repulsive van der Waals forces which control the 3-dimensional structure or the folding of structure. The attractive van der Waals forces involve the interaction among induced dipoles that rise from fluctuations in the charge densities that occur between adjacent uncharged unbonded atoms. So, what is the meaning? Attractive van der Waals forces, it involves the interaction. The interaction is between the induced dipoles that has arisen from the fluctuation in the charge densities and between the adjacent uncharged unbonded atoms. So, van der Waals forces, the attractive force van der Waals forces are formed here. And now, the repulsive, because we have seen that van der Waals forces are both the attractive one and the repulsive one. Now, repulsive van der Waals forces are involved the interaction that occurs when uncharged unbonded atoms come very close to each other. The repulsion is the result of the electron repulsion that occurs as two clouds of electrons begin to overlap. So, again we have to understand there why there is repulsion force and how it is formed. So, repulsion is the result of electron repulsion. So, if, repulsion is between electron repulsion that occurs as two clouds of electrons. If there are two clouds of electrons, they repel each other and they begin to overlap each other, and this repulsive van der Waals forces, they are. So, van der Waals forces are, again there are certain circumstances where attractive van der Waals forces come and there are repulsive van der Waals forces, and both of these van der Waals forces are engaged in the formation of 3-dimensional structure. Now, van der Waals forces, again we are seeing that van der Waals forces are extremely weak and relative to the other forces. We have seen what are the relative forces, they are disulfide linkages, they are hydrogen bonds, they are hydrophobic interaction, electrostatics, salt bridges. These are the forces which give protein a 3-dimensional structure, but out of these van der Waals forces are the extremely weak forces related to the other forces. It is the huge number of such interaction, because these are the weak interaction, the how they are able to stabilize the tertiary or the 3D structure of the protein. So, it is the huge number of such interaction that occur in large protein molecules. So, in large protein molecules they are the number of van der Waals forces are extremely high. So, this huge number is actually responsible to give stability to the 3-dimensional structure, and that make them significant to the folding of protein. 3-dimensional structure means what? It is actually the folding of proteins, and this 3-dimensional structure of protein, mainly the regulatory protein which are involved in the interaction with other molecules, the enzymatic activity. So, for this the folding of proteins is very important, and huge number of van der Waals forces in large protein molecules, they are actually responsible for stabilizing the structure. Otherwise, van der Waals forces are very weak forces. Now, you can see here different type of forces giving protein a 3-dimensional structure. The hydrophobic interactions we can see here, then disulfide bridges. Disulfide bridges occur when cysteine amino acid, we can see here cysteine disulfide linkages. Then we can also see here the ionic bond, formation of ionic bond, hydrogen bonds. So, overall we can see there are hydrogen bonds, hydrophobic interaction, disulfide linkages and ionic bonds which come all together and give protein a 3-dimensional structure. Now, these are the different type of bond which we have stabilizing interaction of tertiary structure. As we know that secondary, tertiary, quaternary structure, they are totally formed by the interaction of certain types of weaker bonds, and these weaker bonds are hydrophobic interactions, then disulfide linkages, salt bridges, van der Waals forces, electrostatic bonds and electrostatic forces. So, these are the different forces which contribute the 3-dimensional structure of protein, or which is also known as tertiary structure of protein. So, with this we come to the end of tertiary structure of protein, and now we are going towards quaternary structure of protein. So, till now we have learned about primary, secondary, tertiary structure of protein. Primary structure of protein is the linear sequence of amino acids. Then coming to secondary structure of protein, this linear sequence of amino acids, they fold, they form hydrogen bonds between intramolecular in the same polypeptide chain, and they form a alpha helical pattern. And if the if side by side two of the polypeptide chain, they are side by side and they form hydrogen bond, in between them they form a beta pleated sheet, and there are certain types of temporary structures, motifs, domains, loops formed during supersecondary structures. Then coming to now this secondary structure of protein, already we have seen that secondary structure of protein contributes towards the structure formation, such as formation of hair, nail, horns, keratins, collagens. So, these structure forming proteins, they are of very significant role, very important in the formation of structure. Now, coming to the 3D structure or the tertiary structure of protein. Now, when a polypeptide chain, that is a chain of amino acids, when it folds and it gives a 3-dimensional structure. Now, this 3-dimensional structure mainly is due to the regulatory function and the catalytic function of that particular protein. So, this tertiary structure is formed by weaker bonds. Weaker bonds, when they come together, they stabilize the structure, the 3-dimensional structure. And now coming to quaternary structure of proteins. So, quaternary structure of proteins means when more than one polypeptide chain is involved and then it interact with each other, they give a quaternary structure of protein. Multiple subunits, we call it as multiple subunits, or we call it as oligomers. So, oligomers, when we are talking about two polypeptide chain are contributing towards a structure, they bind together, they interact together and give a single structure. So, these two polypeptide chains, they may be of similar type or may be of different type. When these two polypeptide chains, they are similar, they are known as homodimer. Dimer means two units, two subunits. If there are two polypeptide chain, then it is two subunits, and sometimes there are many subunits, many polypeptide chains, they form a particular protein. So, there may be multiple subunits, and if the multiple subunits they are of different type, we call it as heterodimer. So, homodimer they are same, and heterodimer the subunits they are different. Now, what are the advantages of this structure? The stability and the regulatory part. Each and every subunits of this quaternary structure had its own specificity, has its own function, and it contributes towards the overall mechanism. So, every subunit had its own recognition, had its own function which is defined. Many proteins contains two or more different polypeptides that are held in association by the same non-covalent forces. Again we are saying that these are the same non-covalent forces, because these subunits they get aparted, because these are the weaker bond and these two subunits can be get separated. So, this all this if there are two or more polypeptides, they come together, they associate with each other and they form non-covalent forces, and that stabilizes the tertiary structure of protein. Now, this arrangement of polypeptide units is called the quaternary structure. That means, polypeptides more than 1, 2 or 3 polypeptide units, when they come together, there are weaker bonds, they are arranged, their arrangements are special, their arrangements are specific. So, this arrangement is known as quaternary structure of protein. Now, when we are saying that when 2 or more polypeptide chain are required, that means the protein it may be of 2 polypeptide chain. If there are 2 subunits, we will call it as subunits. If there are 2 subunits, it will be known as a dimeric protein, dimeric quaternary structure. If there are more than 2 or 3, if there are 3 subunits, we will call it as trimeric structure. And proteins with multiple polypeptide subunits, when proteins there are multiple polypeptides, say 4, 5, 6 or more proteins with multiple polypeptide subunits, they are known as oligomeric proteins. So, whether it is a dimeric, trimeric or multiple polypeptide or the oligomeric protein, this gives protein a quaternary structure. So, oligomeric proteins are composed of multiple identical polypeptide chain or multiple distinct polypeptide chain. Again, if the quaternary structure of protein is composed of more than one polypeptide chain, so polypeptide chain may be identical or maybe not. So, multiple identical polypeptide chain, they can form a quaternary structure, or if the polypeptide chain they are distinct. So, multiple distinct polypeptide chain, they can also come together and they can form a quaternary structure of proteins. Proteins with identical subunits, suppose the multiple identical polypeptides they come together and form subunits. So, these multiple identical subunits they are known as homo-oligomers. If there are more than 2 polypeptide chain, multiple subunits and all the subunits they are identical, then it is known as homo-oligomers. And proteins containing several distinct polypeptide chains, suppose there are the protein is multiple, consists of multiple polypeptide chain, multiple subunits, and these multiple subunits they are that means the composition of amino acid composition are different from each other. The polypeptide chain are termed as hetero-oligomers. So, the oligomeric multiple polypeptide chain, the oligomer protein, they may be homo-oligomers or they can be hetero-oligomers. Now, talking to about the examples, hemoglobin. The hemoglobin we all know it is very common protein, it is the most important example which is given for the quaternary structure of protein. Hemoglobin, it is the oxygen carrier, the oxygen carrying protein of the blood. We know that each and every cell of the body it requires oxygen for its metabolism, for its survival, and the carrier protein here is the hemoglobin for oxygen. And these hemoglobin molecules it is it has 4 subunits, that means it has 4 polypeptide chain. It contains 2 alpha and 2 beta subunits, that means 2 of the subunits they are identical and other 2 are they are identical. So, there are two A2, we call it as alpha 2 and beta 2, that means two alpha subunits and beta subunits, they are arranged in a specific configuration with different type of bonds, and they form a quaternary structure to form alpha 2 beta 2. So, therefore, if there are two different type of polymers here, alpha and beta, two of the chains they are of alpha type and two of the chains they are of beta type. So, hemoglobin is therefore known as the heterooligomers protein. So, this is an example of heterooligomers protein, and the interaction of various bonds like hydrogen bonds, ionic bonds, salt bridges, disulfide bonds, the whole particular chain in a particular geometry, it is very important. If there are two or more than two polypeptides, or we are going to say the oligomeric protein, all the subunits they are arranged according to their function, their geometry, how where these subunits they are going to get settled, how this protein, the quaternary structure of protein is formed, the hydrogen bonds, the ionic bonds and the different type of bonds they hold, the holding of different type of subunit, it is very specific to perform a specific function. So, these bonds they hold in a particular chain in a particular geometry, and thus a functional quaternary structure of protein can be formed. Now, here you can see the quaternary structure of protein. So, the given here is a blue colored and red colored chain. So, just to distinguish that this quaternary structure of protein, if again if the two polypeptides they are of same nature, if they are identical, they are known as homodimers or homomers, and if there are only two polypeptide chain, it will be known as dimers. So, homodimers. If the both of the polypeptide chain they are not identical, they are different, and the protein in the quaternary structure is made up of two polypeptide, then it will be known as the heterodimer. So, you can see the particular, and these two polypeptide chains they are arranged in a particular geometry, which is important. Now, here we can see that quaternary structure adds, what is the significance of quaternary? If there are more than 2 or 3 or 4 polypeptide chain, how this they are managed. So, quaternary structure protein adds stability, how this all the subunits they get stability, how this stability is attained. The quaternary structure of protein adds stability by decreasing the surface to volume ratio, of smaller subunit. So, by decreasing surface to volume ratio, it attains its stability as a ratio of small subunits. Now, here we can see the virus. The viruses they are formed, they contains only the capsid region and the genetic material. Now, the capsid region it covers the viral particle. So, it simplifies the construction of large complex viral capsid and proteosomes. So, it is the quaternary structure of protein which simplifies the construction of large complexes, viral capsid and proteosomes. So, by this the quaternary structure of protein it gives stability by decreasing the surface to volume ratio of smaller subunit. Now, here you can see that there are two different type of polypeptides, and these two structure. Now, these two polypeptides they are arranged, the interchange, there is formation of interchain bond, interchain interaction and in a specific geometric fashion. So, that this quaternary structure of protein becomes functionally active, or functionally the geometry or the interaction of the subunits are like that, it should be functionally active, it may be able to perform the function for which it is. Now, here we were talking about the heterodimer protein. So, heterodimer protein, here we can see there are different 2 alpha 2 beta chain, and this chain containing iron heme particle, 4 heme particle. So, these are the hemoglobin molecule containing iron particle, the oxygen carrier, this protein they take the RBCs which contain the hemoglobin, they take to each and every cell, and they are the oxygen carriers. So, each and every cell should get the oxygen for its metabolic activities for survival. These are the details of the hemoglobin molecule. Hemoglobin molecule contains, it is a globular structure with 4 polypeptide chain. You can see here the RBCs containing the 4 polypeptide chain, 2 alpha and 2 beta and 4 heme group in each. Each polypeptide contain a heme group. So, there are 4 groups, they are the oxygen carrier and the storage molecule. The 4 polypeptide chain consist here 2 alpha and 2 beta. So, this is a classical example of hemoglobin molecule, quaternary structure of hemoglobin molecule. Now, these subunits, we were talking about the geometry of this, how these subunits they are arranged. Now, these subunits they are symmetrically arranged, the cyclic system. So, there are 2 cyclic symmetry, we can see here there are 2 cyclics, 2-fold symmetry and 3-fold symmetry. So, the subunits they are arranged in a very specific matter according to their geometry. So, they are symmetrically arranged. We can see here in the diagram itself the 2-fold and 3-fold symmetry, the cyclic symmetry, they are always in the cyclic symmetry. And also this is the dihedral symmetry. A dihedral symmetry, it is a more complicated type or rotational symmetry. So, dihedral symmetry is also known as the rotational symmetry, and it is generated when n-fold rotation axis and z-fold rotation axis intersect at right angles. When it interacts at right angle, you can very well see in the diagram here. So, dihedral symmetry is formed. So, dihedral symmetry is by far the most common type of dihedral symmetry in a protein. Now, this is another example of quaternary structure of protein. It is DNA polymerase. DNA polymerase is the enzyme which is required for replication of DNA. Now, this DNA polymerase enzyme, it contains several subunits. These several subunits they are arranged by the different type of forces in a specific symmetry, and it gives the protein a quaternary structure of protein. So, the key concept of the structure of protein, tertiary and quaternary structure results from folding of primary and secondary structure of proteins in 3 dimensional. Then tertiary structure of proteins, most proteins tertiary structures, they are combinations of alpha helix and beta pleated sheet, loops and turns. Then larger protein also have multiple folding domains. So, folding of hydrogen soluble globular proteins into the native protein, we can see here minimization of solvent accessibility to the surface, then maximization of interpretation, interchain hydrogen bondings. Then comes the quaternary structure of proteins. So, multiple polypeptide chain, when they come closer, then arrangement of this polypeptide chain and multiple proteins in a symmetrical fashion. We can see here in the quaternary structure of protein. Quaternary structure can play important function in multiple subunits, especially in regulation. So, quaternary structure of protein is very important during the regulatory action of the proteins. So, here we can summarize all the 4 structure of protein: primary, secondary, tertiary, quaternary structure of protein along with their functions. So, quaternary structure of protein is basically involved in the regulation, then some proteins are involved in the structure, then movement, catalytic function, signaling and transportation. So, all these functions are of very significance. So, this is the summary of the structural level of protein: primary, secondary, tertiary and quaternary structure. So, with this we come to the end of the structural organization of proteins. Thank you.